everyone. I am Professor Charu Dogra from Ramjas College, University of Delhi, and I have been delivering various topics about biotechnology, particularly animal biotechnology. We have recently completed the session on the molecular diagnosis of various genetic diseases, where we had discussed about various RFLP, SSCP based methods as well as OLA method or different methods that have been used, the ASO dot blot method that had been used to detect the various genetic disorders prenatally or even in after manifestation. We will be starting with a new topic here, where we talk about the application of recombinant DNA in medicinal field. So, how the recombinant DNA technology, just to review, you understand about the recombinant DNA, where the two DNA from different sources are ligated together, how this recombinant DNA technology can be used or is helpful in case of medical field. In this particular case, we will understand the application of this in medicines, talking primarily about the production of recombinant insulin. So, we will talk about the insulin, that what is insulin, what is the structure of insulin, and that we will go on talking about how the recombinant insulin can be generated and how it can be, the human insulin be produced so as to be given to the patients which are devoid of the presence of this insulin molecules. Now, insulin is an essential protein hormone which is secreted by the beta cells of the islet of Langerhans in the pancreas, which is involved in glucose homeostasis, cell metabolism and mitogenesis. I am sure I am not referring or mentioning about the insulin for the first time. All of you must be aware of this particular protein hormone. It is a very important hormone that regulates the glucose homeostasis. We know the prevalence of the diabetes mellitus as well in globally, which is the disease where the insulin production is reduced. So, when the insulin production is reduced, the glucose homeostasis is altered. So, either the production is reduced because of the insulin sensitivity that decreases, and therefore the glucose homeostasis go haywire. So, when the glucose, so a lot of glucose, it just is not able to enter into the cells and therefore metabolize. So, it alters the cell metabolism, and when there is cell metabolism that is altered, then obviously the production, the division or the various functions that the cell produce, even the mitogenesis is also affected. So, insulin is a very important hormone. It is secreted by the islet of Langerhans in the pancreas, one of the types of cells that is going to the beta cells that is secreting this insulin hormone in the blood. Now, insulin is a peptide hormone which is made up of 51 amino acids with a molecular weight of 5082 and its isoelectric point at pH 5.4. So, these are some of the characteristics of the insulin hormone. The important aspect is that it is composed of two chains. One is called as a B chain with 30 amino acids and an A chain with 21 amino acids. Now, these chains are joined together with the disulfide bonds that is connected via the cysteine residues. So, we already know about that when we talk about the organization or the structural organization of various protein molecules. Then we understand that there is a primary structure, a secondary structure, tertiary and a quaternary structure. So, primary structure is the amino acids, that is a peptide chain that is there. So, we talk about that there are two different chains in here. So, that is the primary structure. Now, how these structures are folded, how these chains are folded forms the secondary structure. So, one chain folded on itself forms the secondary structure. When two or three chains come together, it is called as the tertiary structure, and when these 2-3 chains forms one domain, and then there are different such domains. For example, an insulin molecule is a hexameric protein molecule. So, 6 such combinations will come together to give rise to a insulin chain, and that is a quaternary structure. So, in the folding, this folding occurs because of the interchain disulfide bond which is there. The gene for insulin synthesis is located on chromosome number 11p. So, this is how the amino acids look like. So, this is a 30 amino acid chain and it is a 21 amino acid chain, the B chain and the A chain, and as you can see they are connected together by the disulfide linkages. So, when it folds together the insulin molecule, then it obviously has an N terminal, it has a C terminal. These are the interchain disulfide linkages between the cysteine residues. The A chain also has an intra disulfide linkage formed between the cysteine residues of the chain within itself. So, such kind of folding occurs, and this is a 3D structure molecule shown here, where you can see that there is a chain A and there is a chain B, and then within chain A and chain B these are the disulfide linkages. These are the interchain disulfide linkages. You can see it is a ribbon shape. This is a ribbon loop model that is shown here. So, it is a helical in nature, these two chains are helical in nature, and then you have got these disulfide linkages, and then within chain A also there is a disulfide linkage that is present. So, this is one unit of an insulin molecule, and generally the 6 units come together. So, 6 of these will come together to form a functional or an active insulin molecule which is there. So, this is about the structure of the insulin molecule. Now, insulin was first synthesized as a 110 amino acid chain pre-proinsulin by polysomes. So, what happens now? How does the cell synthesize the insulin? So, insulin is not synthesized, this is the final product. This is the final product that we are talking about, but how does the production occur? So, we have got this gene for insulin where we have an A gene and where we have a B gene. Now, this A gene and B gene are initially joined together by what is called as a connected peptide or a C peptide, and it is a single chain that is first translated. So, insulin is first synthesized as a 110 amino acid chain, and it is, as I said, the final product is a 51 amino acid chain. So, initially this 110 amino acid chain is there, which is called as the pre-proinsulin, and it is produced on the polysome. So, I hope you understand what the polysomes are. A number of ribosomes that are attached to the mRNA forms the polysomes, which causes the translation of the chain of the insulin molecule. Now, proinsulin, apart from the A chain, B chain and a C peptide, also contains a N-terminal signal peptide. Now, what does this signal peptide do? You can refer to the cell biology sessions as well, where the production of the protein, it's going through the endoplasmic reticulum, first the formation because of the polysomes and how they can be loaded, the ones which are secreted, the secretory proteins, how they are entering the endoplasmic reticulum lumen, and then from there how it is passed on to the Golgi network through vesicles, and then from Golgi network to secretory vesicles, and then from secretory vesicles to the surface of the plasma membrane or to the plasma membrane where it can be secreted out. So, that we have already discussed in detail in our cell biology sessions, and you can refer to that, where the function of endoplasmic reticulum and the lectures on function of Golgi complex etc. are taken up. So, in here also what happens is that at first the pre-proinsulin is formed, which contains a N-terminal signal peptide. So, this N-terminal signal peptide loads the growing chain, the peptide chain, onto the endoplasmic reticulum. So, it is formed by the rough endoplasmic reticulum, and this is loaded here, and then it is secreted into the lumen of the endoplasmic reticulum. Now, in there this N-terminal signal peptide is cleaved by the presence of the signal peptidase. So, this pre-proinsulin then forms the proinsulin. So, proinsulin is the B chain connected peptide and a carboxy terminal A chain. So, the processing is done in the Golgi complex, but at first the signal peptide is removed, because the role of the signal peptide is to just direct the growing polypeptide chain to the lumen of the endoplasmic reticulum. So, once it reaches there, then the signal peptide is cleaved by the signal peptidase enzyme, and this chain containing the B chain, a connected peptide, and a carboxyl terminal A chain, so pro-insulin is secreted into the lumen of the endoplasmic reticulum. The N-terminal signal peptide directs the insulin to the endoplasmic reticulum, where it is cleaved off by an endopeptidase, further repackaging in the Golgi bodies and is transmitted through the trans-Golgi network. So, it moves up and then it is incorporated into the secretory vesicles, from which it can be released from the beta cells by exocytosis, for example upon glucose stimulation. So, we have generally talking about, whenever you take a very carbohydrate rich diet, or when there is a glucose spike that is occurring. So, that glucose spike in the blood causes the stimulation so that the insulin is released. So, insulin is released, it basically metabolizes the glucose in the blood so that it does not oxidizes various other organs and causes the damage. But if there is a lot of glucose which is there in the blood, the insulin is not able to combat it, and moreover it becomes insulin sensitive as well. It is not able to stimulate the production of the insulin, and therefore there is an excess glucose in the blood and it causes the damage to various other organs because it is not metabolized properly, leading to the disease of diabetes, which is so prevalent here. So, this is how basically the secretion occur. So, this is just shown here. So, this is what is called as the proinsulin. This is the proinsulin, where the signal peptidase has been peptide has been cleaved by the signal peptidase, and then you can see here that the two chains have been cleaved. The C peptide has been removed as well to give rise to the active final insulin, and again the structural organization has occurred, the conformation where the cysteine residues have been formed. And in here it is showing the at the cellular level how it is happening. So, at cellular level you can see that these endoplasmic reticulum, rough endoplasmic reticulum, this proinsulin is produced into the lumen where the signal peptidase is removed, and then this further packaging, the removal of the C peptide etc. occurred in the Golgi network, and then at the Golgi network it is incorporated into the secretory vesicle, and these secretory vesicles then release the active insulin as well as the C peptide to the exterior of the cells and into the bloodstream. So, this insulin, which is the active insulin, then metabolizes the glucose which is circulating in the blood, thus maintaining or bringing it back to the normal level, the spike that has occurred after a carbohydrate meal or whatever, the glucose spike whatever is occurring. So, this is how the insulin is produced inside the cell and what is the structure of the insulin. Now, recombinant insulin, now again what happened was when the insulin structure was discovered etc., then the recombinant DNA technology was again discovered, invented, and this recombinant DNA technology, the scientists tried to form this insulin molecules because there was a widespread need of this insulin which was there. So, initially it used to be produced in animals. So, animal insulin used to be isolated and then it used to be humanized, because obviously there is a codon disparity between the animals and the human codons which are there. So, it used to be codon optimized even for the use, and it is basically altered, or genetic alteration is done in the animal so that it can produce the human insulin, and then that human insulin is injected into the patients of diabetes, so that because they cannot produce insulin by themselves, they can have it from the outside, and that can combat the excess in glucose and keep the glucose levels in check and avoiding the damage that it is causing. So, this insulin production became a major pharmaceutical industries where the recombinant insulin production was there. So, in 1978, the first recombinant DNA human insulin was prepared by David Goodell and his colleagues of Genentech. So, this was a biotechnology company where the investigator and his colleagues produced the first recombinant DNA human insulin in bacteria. So, recombinant DNA technology we have talked about. So, they isolated the genes for the A gene and the B gene, and then they process them, introduce them into plasmids and introduce them into E. coli, and this E. coli finally gave rise to the A chain and the B chain, which was basically then processed outside the cell, and they were made to be the active insulin that can be then injected into the humans. So, this was the first attempt which was done. Then later on, initially the animal gene used to be done by the company called as the Lilly. So, after they produced the recombinant DNA in E. coli, then the Genentech and Lilly signed an agreement to commercialize the recombinant DNA insulin, and it was done in 1982. The first insulin utilizing rDNA technology, which is called as the Humulin R, the Humulin Rapid, and also Humulin N, which is the NPH, the intermediate acting, were marketed. So, these kind of injections, you know, they started circulating in the market, where this is, for example, a Humulin N injection, which is an NPH derivative, where it is an intermediate acting derivative of the humulin. So, humulin is basically human insulin, and it was produced in E. coli. So, in bacteria, these humulins were produced, and they were then, you know, done at the small scale level, and then they were used in the large reactors, where a large amount of protein can be then produced, and those active proteins were then you know, packaged in these vials, and they can be injected into humans, and therefore the blood, the glucose homeostasis can be maintained, and you know, restricting the damage that is caused by the disease such as the diabetes mellitus, which is there. So, that was the talking about that how recombinant DNA technology. Let us look at the details of the technology. Now, we have already talked about the recombinant DNA technology well in detail, that what it needs is the DNA from two different sources. One is called as a vector molecule that contains that antibiotic resistance, and for the plasmids to be screened, and then the DNA, which is our gene of interest. In this case, for example, the A gene and the B gene, and this A gene and B gene are then cloned in the plasmid molecules, and then they are introduced into the E. coli host, where they can be cloned and propagated, and then the processing can be done of these molecules in vitro for the production of the active insulin. So, this you can see here, this is a very generalized method, where the gene for the A gene or the B gene was cloned downstream to these LAC-Z. So, you remember the beta galactosidase, the lac-operon molecules here, and therefore, you can see that the ligated products are produced, then these LAC-Z genes are cleaved so as to give the A gene as well as the B gene, and then in vitro these two are you know, put together, and the disulfide linkages. So, these kind of reactions are important. For example, cyanoid CNBR treatment, cyanogen bromide treatment is going to remove this LAC-Z gene, and then oxidation etc. Is going to give rise to the formation of the cysteine residues or the disulfide linkages, giving rise to the active insulin molecule. So, this recombinant, this is a very generalized mechanism. Again, you know, we will just look at the detail here, that what happened for the production of humulin. So, as you can see that the two different genes are there, and they can be introduced, they can be ligated together. So, in here again you can see the two different genes are there. Then later on the technology was advanced, and both the genes could be cloned in the same vector molecule. So, in this case they are cloned in the different vector molecules, but then they could also be cloned in the same vector molecules, and then conditions were given so that they can even be processed inside the bacteria only. So, all this kind of changes can occur inside the bacteria, and finally the bacteria can secrete the human insulin. So, that was also done later on, but initially the production was done like this. So, you can see here they have been treated, for example, the plasmids have been treated with EcoR BAMH1, the amplified genes contain the EcoR1 and BAMH1 side, then they were ligated together to give rise to the recombinant DNA molecules containing the A gene or the B gene, and then these recombinant molecules can be selected upon different selection molecules. Now, in here, for example, you can see there were two different host strains that were used, the K12 strain and the other strain that is used for containing the B gene product and the A gene product. So this is a constitutively expressing, so no IPTG induction was there. So this LAC-Z is basically, you know, in frame cloned. This A gene and B gene are cloned in frame to this LAC-Z gene for increasing the production of these molecules, because beta galactosidase or lac-operon is a very well established system in the bacteria, and we know about the alpha complementation assay also. So it will help in the screening as well for the production of these molecules. So in this case, because this K12 strain constitutively expresses this LAC-Z gene because of a you know, the gene which is there, and therefore no IPTG induction is there. In here there is a deletion or a mutation of a molecule which makes it inducible. So this is an inducible LAC-Z gene here, and therefore it can be induced with IPTG, and a lot of production is there. And then you can see both the chains can be retrieved, and they can be first treated with the cyanogen bromide, and then they can be treated with the oxidation method, so that both of them can combine together and give rise to the active insulin. So this is the mechanism, the recombinant DNA technology, how the human insulin can be produced. So these are the active human insulin molecules that can be produced from the bacteria by using the recombinant DNA technology, and these active human insulin can then be processed. So this is showing the downstream process application. In here what they have used is a bacteria in which both A and B genes are cloned together. So when they are cloned together, they have been cultured in large fermenters, because when it is a commercial production, then a large number of cells and large amount of protein is required. So we are not just doing at the lab level, but we are doing at the commercial level for the production of insulin gene. So what we need is the large fermenters or the bioreactors, where basically the culture can be you know, generated. There is a lot of culture which is there, and in here you can see that it is not secreted, but it is present in the inclusion bodies. So entire thing is present, whatever the genes, when they are there, the A chain and the B chain, they are present in the inclusion bodies. Inclusion bodies are these bodies which are formed in either in the periplasmic space of the bacteria, and these inclusion bodies will need to be solubilized to purify the protein molecules which are there. So first of all, these inclusion bodies are then isolated, these are differently washed, and as you can see, then there is a chain which is present, the two chains that are present, which are then given the conditions so as to have the active insulin molecule. So you can see first of all that pre-proinsulin is there, then the cleavage is occurring, and you can have the final two chains, and where the further processing is done so as to give the commercial production of the human insulin molecule. So these things are very, very important, and then in these vials the human insulin protein molecules are there that can be stored at a required temperature, the protein, you know, so in cold conditions, and then they can be transported, and you know, they can be marketed, and they were sold. So it's a huge market. The production of the human insulin is a huge market because of the occurrence and even increase in the patients of diabetes which are there. Now last point is that I would just like to mention here that what we have discussed is the basic recombinant DNA technology method, where the human insulin production is done in bacteria or E. coli. Now this method has a lot of limitations, and therefore a lot of advancements came into being, and now the human insulin, there are transgenic plants that can produce human insulin, or there are yeast systems that can produce human insulin, and you know, there are different systems that have been developed so that the insulin production could be there. Now the major limitations was that the plasmids could be lost. So when the external plasmids are introduced into the bacterial host, because they are not their natural plasmids, they have a tendency to cure these plasmids or lose these plasmids. The major limitation is the loss of plasmids, then the development of antibiotic properties as well. Now because these plasmids contain antibiotic markers, and when this is basically introduced, they can develop certain kind of antibiotic properties, which is again not very good. Then there is a lack of post-translational modifications. Now this is again a very, very important limitation which is there, that needs to be understood, and therefore the genes were tried to be cloned and expressed in higher organisms. Now in bacteria there is no post-translational modifications that occur, and therefore that has to be done in vitro. So if we have higher organism system where the post-translational modifications like the eukaryotic system is occurring, then it is easier to produce the human insulin in the form that it is produced in the humans. Then as I said, that the inclusion bodies accumulation within the cells is a bit problem, because then we have to isolate the inclusion bodies, and when there is a lot of inclusion body accumulation with the production of the foreign proteins, then the cell death can also occur. There is a tendency of improper folding, because it is not a eukaryotic host, it is a prokaryotic host, so the folding system can differ, the proteolytic digestions could be there, and again there could be poor secretion that makes the protein recovery difficult. So there are many limitations which are there for the production of human insulin in E. coli, and therefore many advancements occur for developing the system in yeast, or developing the system even in plants and other higher organisms like porcine insulin and all those kind of things, where the higher organisms were used to produce the human insulin. So this was one of the molecule that we discussed today. In the next session we will talk about the recombinant growth hormone production, and then we talk about the gene therapy, which would end our series as far as the NEP syllabus is concerned regarding the biotechnology that we have been continuing for some time now. Thank you.